Hello and welcome to the instructor lecture for Biology 1308. What I would do before you even start going through this lecture is I'd have your lecture handout available. You can have that available online in the PDF format, or you can download it in Word and take notes directly, or you can print it out. But I would definitely have that handout out and available to you and be following along with those prompts as we go through this so you can think about the material and think about how I'm going to formally evaluate you on the material. The other thing I would keep in mind is I've chosen the lectures that I'm giving you in video format rather than module format because these things tend to be problematic in AMP1, and I want to highlight these particular topics because I don't want you to be steamrolled when you get into AMP1 and you hear these terms and you're introduced to these topics again. I want you to have an idea of the depth and breadth that you're going to have to understand some of these things in AMP1 and how it translates over into AMP1, and that's why I've chosen specific topic points. So the quiz associated with my lectures as opposed to the modules, my lecture quiz is worth 50 points, whereas the module quizzes are worth 10 points. So I think that's important to highlight and important for you guys to know. So the lecture that we're going over today is the plasma membrane, and we're going to be drawing a focal point to passive transport. You have your learning objectives, you have your handout out and available to you, so let's go ahead and get started. Key topics for this lecture are going to be talking about the domains of life and being able to distinguish the domains of life. Then we're going to talk about membrane structure, and we're going to draw a focal point to the plasma membrane. And finally, we're going to talk about passive transport, and then we're going to get into diffusion and osmosis, tonicity, and how that relates to IV science, which is really why we're studying this as a class to prepare you for AMP1 and ultimately to prepare you for allied health. So when you think about domains of life, there are three major domains, two that we're going to focus on in this class. You have eukarya and prokarya, or eukaryotic cells or organisms and prokaryotic cells or organisms. When you think about eukaryotic organisms, here we have a plasma membrane, and what we're looking at are the internal contents of the cell, and we see the internal contents of the cell are compartmentalized by these little things called membrane-bound organelles. If you see membrane-bound organelles in a cell, you know that cell is eukaryotic. By definition, that's the first major division of life when you think about phylogenetics, how we categorize life, right? You have eukaryotes and prokaryotes. Eukaryotic organisms are made of cells that have membrane-bound organelles. Prokaryotic organisms do not. So the only thing you need to see about a cell to know for a fact that it's eukaryotic is a nucleus. So a lot of times under a light microscope, you're not going to be able to see all these little membrane-bound organelles over here, but you're almost always able to see the nucleus because it's dense, it's kind of centrally located, it's a very large structure, right, relative to the overall cell size, so you tend to be able to see those. So if I ask you which of the following would be true of the cell depicted above, or the image of the cells above, or the image of the tissue above, and you're debating between eukaryotic and prokaryotic, if it has a nucleus, right, it means it has other membrane-bound organelles and it is eukaryotic, as opposed to prokaryotic organisms like bacteria, which do not have membrane-bound organelles. They have membranes, right? So you have a cell wall followed by a cytoplasmic membrane, and that membrane is very similar in structure to the membranes in eukaryotic cells, but if you look internally in the cell, there are no membrane-bound organelles. You have this diffuse grouping of nucleic acids with ribosomes kind of peppered around and proteins all over the place, but there's no compartmentalization, right? There are no membranes separating like a nucleus from a mitochondria from a Golgi apparatus or whatever it is. Now, another thing I want to highlight, so animal and plant cells are eukaryotic cells. You can tell that because they have these membrane-bound organelles which allow for specialization of function. Another thing that I want to highlight about prokaryotes, when you think about prokaryotic organisms, what we're really talking about are bacteria, right? When you use the term prokaryote, you're really talking about bacteria. Another thing I want to highlight is prokaryotic cells tend to be much smaller than eukaryotic cells. So if this was let's say a human skin cell, or whatever it is, right? If you were to compare this bacteria next to it, that bacteria would look like that E right there. It's significantly smaller. Prokaryotic cells are significantly smaller than eukaryotic cells, much, much smaller. So it's important to kind of highlight that. So if I was to ask you a question on an exam, and what would you do in anatomy and physiology? In the first unit, you look at what's called tissue histology. And histology, remember, when you think about groups of cells working together to carry out a common set of functions, you have a tissue, right? Histology is the study of tissues, and it forms a centerpiece of anatomy and physiology. It's one of the first things you go over. And when you're looking at these cells here, if I was to ask, how would you categorize the cells above? Well, here's a nucleus, here's a nucleus, here's a nucleus. Those would definitely be eukaryotic cells. And then those cells come together to form tissues, and you're going to have to learn, not in this class, but in A&P1, big, long, convoluted sounding terms like pseudostratified ciliated columnar epithelium, right? You at least have the background to go, okay, eukaryotic cell, right? All cells that have membrane-bound organelles. If I see a nucleus, I know it's eukaryotic. We are eukaryotic organisms. Humans are eukaryotic organisms. If an instructor says it's difficult to target eukaryotic pathogens like disease-causing agents like tapeworms because their cells are similar to ours, you know, oh, right, if you're throwing in a medication that kills cells with membrane-bound organelles, we may kill our own cells as well. So it's difficult to develop those medications. It's not as easy to find targets. So terminology, and this is kind of with your follow along, we have cytology, which is the study of the cell. And we have pathology, which is the study of disease. If you ever see cyte in any word root or any word, cyte is a word root that means cell, and ology is the study of. And pathology is the study of disease. So cytology, study of the cell, pathology, the study of disease. Now, why do I mention these two together? Oftentimes, what pathologists do is they utilize things like general chemistry with respect to evaluating chem panels, biochemistry, looking at different biological markers in the blood, cell biology, tissue histology. A lot of times, pathology, which is kind of the centerpiece of the study of disease, the focal point is not at the macroscopic level, the things you can see with the unaided eye. A lot of times the focal point is at the microscopic level, right? And the molecular level, the very small things, because that's often where the changes associated with disease really begin. So if I was to give you a question and I said a pap smear is a procedure in which a doctor takes a probe in order to scrape off cells in the cervix to look at them under a microscope, this would be an example of, that would be cytology, right? And if you say those cells, when you look at them under the microscope, changes in those cells can be indicative of cancer, that's how cytology can play into things like pathology. That's what pathologists do, essentially. They look at these very smallest components to look at markers of disease. Now, one of your follow-alongs asks, what is a stem cell? So we're getting into cell biology. We're going to be talking about the plasma membrane today, and then, in your module, you're going to go over organelles. And you think about what cells are. Well, cells are the building blocks of organisms. We are made from 200 trillion cells put together in specific geometric arrangements peppered throughout our body. We have 206 different cell types that serve a variety of functional roles, from firing electrical signals and allowing for communication to contracting in order to generate force, to absorbing things along the GI tract. We have 206 different types of cells, right? Clustered together, working in unison to carry out a mind-bogglingly complex array of functions. All of those cells that we have in our body, all of those adult cells that we call somatic cells, ultimately derived from what are called embryonic stem cells. Now, embryonic stem cells and stem cells in general are cells that do not have a defined function. So when you look over here, what we're looking at is the division of cells and then the growth during embryonic development of a developing human being, right? So we have this embryo, week 0 to 8 is the embryo, and what you're seeing is this massive division of cells and this specialization of cells to serve different functions. So from that single cell, it underwent a ton of different divisions, and then those cells started to specialize, and some of those cells became brain cells, some of those cells became skin cells, some of those cells became liver cells. So embryonic stem cells, when you get them like right there in development, these stem cells haven't been told what to do yet. They don't have a defined function. So stem cells don't have a defined function yet, so we say they're non-differentiated. They do not have a function yet. They are non-differentiated or undifferentiated cells. That's another way of saying it. Now, the beauty of stem cells is stem cells, if they get the appropriate chemical cues, can become anything. So if you give the appropriate chemical cues to these stem cells, they can become neurons that make up your brain. They can become the cells of the pancreas that produce, insulin or glucagon. They could become skeletal muscle cells that contract to generate force. So stem cells are undifferentiated cells. And through the process of embryological development, you have these cells and they get chemical messages, and those chemical messengers tell those cells what to become. So when a stem cell becomes a specific type of cell, like a skeletal muscle cell, we say that that stem cell has differentiated. So when a cell takes on a defined function and it changes its shape and its metabolic characteristics and its functional characteristics, we say that cell has differentiated. The reason that stem cells are so valuable in medicine is they hold a lot of promise for things like tissue engineering, wound healing. If you have a spinal cord injury, for example, and those cells are no longer dividing, and you introduce stem cells and you figure out how to get them to divide to produce the neurons to maybe heal that injury, that's a lot of potential that stem cells have with respect to breakthroughs in medicine, and they're already massive breakthroughs in medicine as a consequence of understanding stem cells. So stem cells are non-differentiated. When they mature into an adult functional cell, we say that they have undergone the process of differentiation, and then they serve a specific functional role. And we have 206 different types of cells. Some are big, some are small, some you find along the GI tract, some you find in your brain, but all of those cells working together literally make up you. So stem cells, adult or somatic cells. Undifferentiated, differentiated cells. Undifferentiated, no defined function, and they can become anything. That's why they're so valuable from a clinical perspective. Differentiated, they've now matured and take on a defined function, and we consider those adult cells after the differentiation occurs. Then you're talking about things like skeletal muscle cells or neurons or whatever, but all the cells in your body ultimately derive from stem cells at one point in time. Now, a traditional representation of a cell is a diagram that looks kind of like this. This cell doesn't actually exist anywhere in biological systems, but it's a useful diagram for understanding abstract concepts related to the cell. So we don't actually have this cell anywhere in our body, but this cell, is a useful way to go, okay, here's a cell, here's the internal machinery, that's what the internal machinery does. Now, in your module, you're going to be talking about the different organelles of the cell, and then in the next lecture I give, in the next unit, we're going to talk about the nucleus and the rough endoplasmic reticulum and the processes of transcription and translation, but that's not the focal point for today. So you have a cell, and it's a eukaryotic cell because you get these membrane-bound organelles, which allow for compartmentalization and specialization of function. So they allow the mitochondria, for example, to work independently from the Golgi complex or the Golgi apparatus, and these two different pieces of machinery in the cell have unique functional characteristics, and they're allowed to do that because they're compartmentalized. They have their own little space. That's the beauty of that. Now, these membranes that surround the organelles internally, kind of forming the border of the organelles, we refer to those as cell membranes. So when we say membrane-bound organelles, here we have the cell membranes around the rough ER, the smooth ER. When I use the term plasma membrane, the plasma membrane, even though it's structurally similar to these cell membranes, the plasma membrane is the large membrane that forms the barrier between the outside and the inside of the cell. So if I say a cell membrane, I'm talking about an internal membrane within the cell, probably around a specific organelle. When I talk about the plasma membrane, I am talking about the primary membrane that forms a barrier, a selectively permeable barrier between the outside and the inside of the cell. So Just keep that in mind. Now, this cell diagram I've thrown up there would probably be useful. I'm going to use it again when we talk about organelles on the exam and when we do our exam review. It's not a bad diagram to know because it just kind of condenses a lot of seemingly disparate information into here's a cell, here's the machinery, here's the functional roles of those machines in the cell that we call organelles. Now, for this lecture, our focal point is going to be on the plasma membrane, this large membrane that forms the barrier between the outside and the inside of the cell. And one of the terms I'm going to be using quite a bit, and we're going to tie it to anatomy and physiology, is I'm going to be using extracellular fluid, or ECF, and intracellular fluid. Extracellular fluid is a collective term for all of the fluid outside of the cell in a living system. Intracellular fluid is a comprehensive term for all the fluid inside the cell in a living system. So, for example, you're going to read about the cytosol being that kind of aqueous solution inside the cell. The cytosol and the intracellular fluid, when you say ICF, you're talking about the collective fluid inside all the cells in the organism, right? Now, if you're talking about a single cell system, that's just used to represent what's happening at a larger level, because we never really care about just an individual cell. We're tying this to what's happening in the entire organism. So intracellular fluid is a blanket term for all the fluid inside cells. Extracellular fluid is a blanket term for all fluid outside of cells. Now, when you think about extracellular fluid, right, I want you to understand where that comes into play in the body. So you have your cardiovascular system, and the job of the cardiovascular system, which consists of your heart, blood vessels, and the blood that flows within them, is to move material around the body. So the body's constantly moving material around. We want to deliver things like oxygen and nutrients to the cells of the tissues of our body, and we want to get rid of things like carbon dioxide and metabolic waste. So what the heart does is the heart's a hydraulic pump, and when it contracts, it physically moves blood up and out to a series of tubes called blood vessels. The blood vessels that take blood away from the heart are called arteries. That blood flows away from the heart. Those arteries branch and branch and branch over and over again until they form these very small blood vessels that allow for exchange to occur called capillaries. After that blood flows through the capillaries, we deliver oxygen and nutrients to tissues at the level of the capillary, and we take away carbon dioxide and metabolic waste at the level of the capillary. Those capillaries are then drained by veins, right? And those veins bring blood back to the heart so that process can start anew, and we're constantly moving stuff around. So when you're looking here, what you're looking at is a blood vessel with a plaque forming, and that's not the important point here, but what you see are those platelets catching on that plaque and contributing to that plaque getting larger and larger, which is going to eventually cause problems with that blood vessel. But what we're really looking at here is you have blood flowing, so the heart pumps and it propels blood through these things called blood vessels, and we've all kind of seen an image like this. Now, here we have red blood cells, here we have platelets. On this particular GIF, you don't see any white blood cells, but that's not the point here. Notice that those red blood cells and platelets are floating, right? So there has to be fluid outside of those cells, and that fluid that you find outside of those cells is a form of extracellular fluid called blood plasma, right? So when you look at these cells, they're floating in blood plasma, and blood plasma is an aqueous solution, a water-based solution with tons of stuff like nutrients, ions, chemical signaling molecules, waste, and that's how we move stuff around in the body. So you have blood plasma, which is one type of extracellular fluid. At the level of a capillary, think of a capillary being kind of like a hose. And if you poke holes in that hose, right, and you turn on the water and you give a water pressure, some of that water is going to flow out of the hose. It's going to flow from the lumen, or the inside of the hose, to the outside of the hose. Well, a similar process takes place at blood capillaries. Some of the fluid in blood capillaries, specifically the blood plasma, gets pushed across the capillary wall, and it moves into this space surrounding your cells. This space surrounding your cells is called the interstitial space. And the moment that plasma exits the cardiovascular system and enters that space surrounding your cells, it's no longer called blood plasma, it's called interstitial fluid. So that blood is delivering things like nutrients and oxygens. That plasma gets pushed across into this interstitial space where it becomes interstitial fluid that actually bathes the cells in your body. So interstitial fluid is another form of extracellular fluid. This isn't just empty space, right, between these cells that make up the tissues of your body. Eventually, that fluid gets drained by what's called the lymphatic system. So you can't have fluid continue to build up forever, for example, in your hand. Otherwise, if that interstitial fluid just continued to build up, you would have a swollen hand or you would develop a condition called edema, which means accumulation of fluid. So you need a way to drain that fluid, that interstitial fluid, and that's what your lymphatic system does. In fact, that's the primary function of the lymphatic system, is to drain excess interstitial fluid. When that fluid moves from the interstitial space into the lymphatic capillary, that fluid is now called lymph. So there are three types of extracellular fluid to be familiar with: plasma, interstitial fluid, and lymph. The most important are plasma and interstitial fluid for the discussion that we're going to be having. So when you look at your total body mass, it varies depending on muscle composition, adipose composition, etc. There are a lot of variables here. But in general, right, women about 45% solids, men about 40% solids, and then women about 55% liquid, men about 60% liquids, right? Water, essentially. Then we look at how those fluids are broken down in the body. So collectively, because our body is made from like 200 trillion cells, we're physically made up of cells, we have way more intracellular fluid than we have extracellular fluid. About 2/3 of all the water in your body exists inside of cells, and about 1/3 exists outside of cells. So about 2/3 of your total body fluid is inside cells, and about 1/3 is outside of cells. So we have significantly more ICF than we do ECF. Remember, we're talking about the fluid inside all of the cells. So we have more intracellular fluid than we have extracellular fluid. And then when you break extracellular fluid down, you can break it down into blood plasma and interstitial fluid. We have way more interstitial fluid than blood plasma. At any given point in time, you have about 5 liters of blood. About 2 liters of that blood are red blood cells, platelets, and white blood cells, and 3 liters of that blood, which is a mixture, is blood plasma. So at any given point in time, we have about 3 liters of blood plasma, and we have about 12 liters of interstitial fluid. So a lot of people, that's counterintuitive to, but we have way more interstitial fluid than we have blood plasma. So this is kind of the breakdown of body fluids and why we talk about things in generalities like ECF and ICF, particularly when we're talking about IV science. Now, that leads us into talking about the plasma membrane. Now, in your previous modules, you've gone over the plasma membrane and you looked at membrane transport, and we're going to highlight some of those things here before we get into tonicity, osmosis, and why we care about it. So when you think about the plasma membrane or any of the membranes in cells, you have the structural components of the membrane. And the structural components of the plasma membrane are really phospholipids and cholesterol. And we're going to take a deep look at both of these in just a moment. The functional components are proteins. Remember, proteins, even if we draw them as blobs, remember that they are amino acids linked together covalently to form peptide bonds that fold kind of like origami to form machines. Proteins are little molecular machines. They're tiny little machines that do stuff. And some of the proteins, right, that we code for with our genetic code, our DNA is really just information for making proteins. Some of those proteins exist embedded within cell membranes. In this case, the plasma membrane, and they form the functional components of the membrane. So they can be things like transporters, channels, receptors, cell surface identity markers. So as we go through this, and we're talking about cell membranes, one of the reasons we have membranes internally in the cell, and I've mentioned this a few times, but it's worth noting one more time, is compartmentalization. So if you look at a house, for example, you have the living room, the kitchen, the bedrooms, the bathroom, and what you're seeing here is what's called compartmentalization. So the kitchen has certain equipment and is responsible for carrying out certain tasks. The living room has certain equipment and is responsible for carrying out certain tasks. So what you see here is that the kitchen is specialized to serve a function, in this case cooking and storing food. The living room is specialized to serve a function, in this case seating and entertaining people. The bathroom is specialized to serve a function, right? To shower and use the restroom in because it has the machinery to do it. And that's what compartmentalization allows in the cell. It allows for specialization of function. So the nucleus, right, is separated and therefore it can function, it can have its own unique set of functions independently from the roughy R, independently from the Golgi apparatus, independently from the mitochondria. And they communicate and they work together, but compartmentalization allows for specialization of function, and that's what cell membranes do. Our primary membrane, the plasma membrane, is structurally and functionally similar to the membranes internally in the cell, but this big daddy on the outside we call the plasma membrane. Now, this plasma membrane, the best description of it is it's a selectively permeable membrane, right? That's made up of lipids and proteins. Selective permeability refers to the fact that this membrane will allow some stuff across, but it won't allow other stuff, hence the term selective permeability. And we're going to talk about that after we talk about the structure of the plasma membrane. So if you were to look at the plasma membrane, what you'll see is most of it is made up of what's called the phospholipid bilayer. And it's called the phospholipid bilayer, one, because it's made up of phospholipids, and two, these phospholipids arrange themselves in a double layered structure based on the chemical characteristics of the different parts of the phospholipids, and that's why it's called the phospholipid bilayer. Now, if you notice, here we have our extracellular and our intracellular fluid. And when you look at a phospholipid, you have the head of the phospholipid and then you have two fatty acid tails. The head of the phospholipid contains a polar phosphate group. In other words, the head of phospholipids is hydrophilic. That means it'll be attracted toward water. So notice the position of these phospholipids with respect to the extra and intracellular fluid, which are really water-based solutions. So those heads orient themselves toward that water-based solution because they're attracted to the charge on the water molecule. So the polar or hydrophilic phospholipid heads will always orient themselves toward water, and these non-polar, aka hydrophobic fatty acid tails, can't orient themselves toward water. The water is repulsive, so when you mix oil and water, they separate because there's no basis for interaction. So what you'll notice, and this goes back to the likes dissolve likes, is that when you think about solution chemistry, these phospholipids, or these fatty acid tails on these phospholipids don't interact with water, but they interact with one another. So on an exam, like if I was giving you short answer questions, one of the short answer questions I always give to my students is I say, hey, you have a handful of 8 large phospholipids and you throw them into water. How will they spontaneously arrange themselves? And why is that important with respect to the maintenance of the integrity of the phospholipid bilayer? Well, they'll spontaneously arrange themselves into a bilayer, this bilayer, in order to minimize the interaction of the head with the lipids. You want the head's going to orient always toward water, and the fatty acid tails are always going to orient toward one another. So what causes the formation and what maintains the structural integrity of that phospholipid bilayer has to do with these hydrophilic heads and these hydrophobic fatty acid tails, right? The fact that the hydrophilic heads will always orient toward water and the hydrophobic tails will always orient toward one another, that's what maintains that structural integrity, the interaction of the phospholipids with the aqueous solutions and with one another. Now, shoved up in between these phospholipid bilayers, you get this little molecule here called cholesterol. Now, cholesterol is a lipid-soluble molecule. You can see the 1, 2, 3, 4 rings forming the steroid ring. And what cholesterol does is it kind of gets inserted into the phospholipid bilayer where it imparts a little bit of structural integrity. So it reinforces the phospholipid bilayer. And what it does is it really prevents phospholipids from clumping too close when it gets cold or from flying apart when it gets hot. Cholesterol is also really important in stabilizing things like functional proteins. So, whereas you have your phospholipids and your cholesterol molecules, which make up the primary structural component of the cell membranes, or the plasma membrane in this case, all of these big guys here are proteins, right? So you have protein, 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 protein. This is a protein pinned to a bunch of glucose molecules. So what you're looking at here is a glycoprotein, which really means just sugar pinned to a protein. So you'll hear those terms used. This is another glycoprotein, sugar pinned to a protein, or these glucose molecules, these polysaccharide chains pinned to this protein. So now we're kind of oriented to the phospholipid bilayer. If I show you an individual phospholipid, I would never expect you to draw out an image like this. But it does give you some interesting information when you look at this. Like here you have your polar phosphate head that carries that negative charge, and that's what allows those polar heads to interact with water. And you have these nonpolar fatty acid tails. And we should know at this point that this is not just a squiggly line. What we're looking at is a line angle formula of a fatty acid. So this fatty acid, for example, would have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17 proteins. Or, sorry, 17 carbons, right? Remember, this is just a long chain of carbon and hydrogen. This one, you could do the same thing and you can see that it's an unsaturated fatty acid. But what you're looking at are hydrocarbon chains right here, and they're just drawing it like that to make it easier to understand. Now, if I was to ask you to draw a phospholipid, you could use this line angle formula to indicate that you understand that fatty acids are really just long chains of carbon bound to hydrogen. Or you could draw a picture kind of like this, where you have the hydrophilic head and the hydrophobic tails. Remember, there are multiple ways to say hydrophilic and there are multiple ways to say hydrophobic. For example, hydrophilic could be polar, right? So polar and hydrophilic mean the same thing, as does water soluble, as does lipid insoluble, as does charged. So you'll notice that there are multiple ways to say attracted to water or soluble in water. Same thing with hydrophobic. Hydrophobic molecules are nonpolar, aka water insoluble, aka lipid soluble, aka non-charged. So when you think about those things, there are multiple ways of saying hydrophilic and hydrophobic. The most important for this class is you could say the hydrophilic slash polar head and the hydrophobic slash non-polar tails, because those polarity characteristics are important. Does it carry a charge or doesn't it? Now, the cholesterol, you should recognize cholesterol at this point, and the only steroid I'm going to give you on this exam is cholesterol. Cholesterol shoves up into the phospholipid bilayer, providing a little bit of structural integrity to the membrane, right? Cholesterol, when it enters the cell and it's used, for example, in some metabolic pathway, cholesterol is also really important in synthesizing steroid hormones. But for this exam, focus on the fact that cholesterol is a critical structural component of cell membranes because it shoves up into those phospholipids and stabilizes those phospholipids internally. Now, we've talked about the lipid portion, right, of the plasma membrane and of cell membranes in general. Now let's talk about the proteins. So lipids form the structural component. The primary structural component are these phospholipids. So you get those polar phosphate heads that orient themselves toward the extracellular fluid and intracellular fluid because they're hydrophilic, polar slash hydrophilic, right? They energetically, it's most favorable for them to be oriented toward water. And then you have these nonpolar or hydrophobic fatty acid tails that orient toward one another. And it's the interaction of the phospholipids with water and with each other that ultimately maintain the structural integrity of the phospholipid bilayer. So if you're labeling, for example, you have your phospholipid bilayer and then you have these hydrophobic slash nonpolar tails, you have these hydrophilic slash polar heads, and I say, which of the following would best describe, or which of the following would best explain what maintains the structural integrity of the phospholipid bilayer? Well, the structural integrity of the phospholipid bilayer is maintained by the interaction of the phospholipids with water. Specifically, these polar phosphate heads, right, interact with the polar water molecules, so they always orient toward water. And these nonpolar fatty acid tails, or these hydrophobic fatty acid tails, always orient toward one another. So if you put phospholipids into water, they will spontaneously form this bilayer without any kind of energetic input, and that's really important in living systems. Then you have the functional components of the phospholipid bilayer, and those are proteins. So remember that even when you draw a blob model of a protein, and these proteins, particularly like channels and transporters, become a very important theme in anatomy and physiology, when you're looking at this blob model of a protein, understand that it is a structure made from amino acids that folds into this kind of channel structure, and we're just drawing it as a blob for the sake of simplicity. So you have this blob model of a protein, and channel proteins are really just molecular donuts in the cell membrane that allow small polar molecules across the cell membrane. Channel proteins can be closed or they can be open, right? So they can open and close, but when they're open, they exist in an open state, and when they're closed, they exist in a closed state. So what we're looking at here is an open channel protein. Now, channel proteins, a really important functional characteristic of channel proteins and carrier proteins is that they're specific. So if I say I have a sodium channel, that sodium channel will only move sodium. If I say I have a potassium channel, that potassium channel will only move potassium. If I say I have a calcium channel, that calcium channel will only move calcium. So one of the really important characteristics of channel proteins is that they're specific, and they transport small polar molecules like ions, sodium, potassium, chloride, magnesium. And these channels are really, really important to understand what a channel protein is, because in A&P you hear about it all the time. Another type of protein is a carrier protein. Now, carrier proteins don't exist in an open or a closed state. The carrier proteins need to bind to their solute, aka their substrate. And when they bind to their substrate, that causes a change in the shape of the protein that ultimately moves that substrate across, or that solute across, the membrane. In this case, we'll say the plasma membrane. So carrier proteins don't exist in an open or a closed state. That solute needs to bind to that protein, and that protein will change its shape, but it never just allows just the free flow of material across the cell membrane. The reason that carrier proteins don't exist in an open or a closed state, that you actually need to activate them, and they prevent the free movement of solute. One, carrier proteins are specific. So what I want you to know is that carrier proteins move large polar molecules. So large charged molecules like sugars and amino acids. That's how, for example, glucose gets into the cell. It binds to a carrier protein, and that carrier protein changes its shape and kicks that glucose molecule into the cell. So they bind larger polar molecules like sugars and amino acids. And the reason that they don't just exist in an open or a closed state is if this carrier opened, you would have all sorts of solutes flowing through, because that would be a big gap in the cell membrane, and you don't want that. So they move larger polar molecules across the cell membrane, and they're really, really important when you get into anatomy and physiology. So you have channel proteins, you have carrier proteins, then you have what are called receptors. So a ligand is a blanket term for a signaling molecule. When you see the term ligand, that's a blanket term for a signaling molecule. The ligands that come up most often in anatomy and physiology are hormones and neurotransmitters. So when you see the term ligand, that's a blanket term that just means chemical signaling molecule. And the chemical signaling molecules, or the ligands we care most about for anatomy and physiology, are hormones and neurotransmitters. So that's what you're going to focus on. So this could be either a hormone like insulin, or it could be a neurotransmitter like epinephrine or norepinephrine, or glutamate or GABA, whatever it is. So these ligands, right, these cell signaling molecules bind to receptors. And what receptors are proteins embedded within cell membranes. In this case, we have the plasma membrane. And what they do is they communicate. They form a point of communication between the outside and the inside of the cell, because they have what's called a transmembrane spanning property, meaning they have portions of the protein outside of the cell and portions of the protein inside of the cell. So when a signaling molecule binds to a receptor protein, A message that was outside of the cell becomes events or changes inside or internally in the cell. So, a really common class of receptors are what are called G protein coupled receptors. And you don't have to know this pathway, but when a ligand, for example, a hormone or a neurotransmitter, binds to a G protein coupled receptor or a GPCR, that triggers the activation of a G protein, which activates adenylyl cyclase. Adenylyl cyclase then converts secondary signaling molecules, specifically ATP, to cyclic AMP, right? Cyclic AMP essentially turns proteins on, like protein kinases. These protein kinases then exert some kind of cellular response. So, from a message that began outside of the cell, right, you get a set of intracellular responses, and that's what receptor proteins really do. Is they form a point of communication between the outside and the inside of the cell. So, receptor proteins, again, are really important. You'll see a lot of diagrams like this where you see the ligand, either the neurotransmitter or the hormone, binding to the protein receptor embedded in the membrane. That activates what's called a signaling cascade inside the cell, which is going to produce some kind of cellular response. So when you think about the types of diagrams you're going to get, here we have what's called dopaminergic synapse. So what you're seeing here is the release of dopamine. And dopamine is a molecule that makes us feel really good. It's a reward molecule, right? It's part of what's called our mesolimbic dopamine circuits, which are our reward circuits. So things like opiates bind to, they trigger the release of dopamine, and that's what feels so good about them. So when you think about medications to treat things like opiate overdoses, what you're talking about is receptor physiology. So here we have an opioid receptor, which is going to trigger the release of dopamine. And naloxone is essentially, you can have injectable naloxone, you have nose spray naloxone. That binds to and competes with that opioid, preventing that opioid from doing what it's preventing that opioid from working at the level of the receptor, which prevents the release of dopamine, and it can be very, very useful, for example, in overdoses. So you're talking about receptor physiology. What naloxone does is it essentially blocks opioid receptors, preventing them from exerting their cellular response and upregulating the release of dopamine. So finally, with respect to functional proteins in the cell, we have what are called major histocompatibility complex markers, or MHC markers. MHC markers are self markers. So the way that the immune system works is white blood cells float around the immune system and they look to see self and non-self. Right? So the reason that your immune system doesn't trigger a massive reaction and destroy all the tissues in your body is because the cells that make up your tissues have these little markers of self on them. And these white blood cells come around and they see these little protein markers, which are genetically unique for every individual, and they go, oh, that MHC or that protein marker, which is like cellular ID, that's fine. I'm not going to destroy that cell. But if you have a cell or something in your body that doesn't have the appropriate marker, right, like let's say this bacterial cell, that white blood cell is going to recognize that that protein marker is off, and it's going to now characterize this molecule as being part of non-self. When the immune system recognizes non-self, that's what triggers an immune response. So this particular white blood cell is now going to target this bacteria, gobble it up, and destroy it. So that's how the immune system works, right? We identify self from non-self, and we do that using these protein markers called MHC markers, which are embedded in every cell in your body with the exception of your red blood cells, and we'll won't get into that in this class at all. So this white blood cell is coming over and it's probing and it's going, okay, that's fine. We'll leave that cell alone. That cell is part of self. As opposed to, let's say the same cell becomes infected with a virus like the coronavirus. What do you notice happened to this cell in response? So this cell has a response in which it removes those MHC markers from its membrane. And now this cell can't recognize the infected cell, so it's going to destroy that infected cell because it no longer has the ID necessary to keep these white blood cells happy. So when you think about this, MHC markers are like little tags that have your name genetically, right? So let's say that we have a liver cell, and this liver cell belongs to somebody named Ashley. When you think about this conceptually, a major histocompatibility complex could be like a little marker or a little tag that says this is an Ashley cell. And if this liver cell, which is an Ashley liver cell, exists within Ashley, it's going to have Ashley white blood cells. And Ashley's white blood cells are going to come around and they're going to probe this cell and they're going to see, okay, right, that cell's supposed to be here. It's part of self. I'm not going to worry about it anymore, and I'm going to move on and continue with my surveillance. It's going to give you the thumbs up. They don't really look like that. That's just a cartoon depiction. Now, when you think about the physiological basis of rejection, organ rejection during organ transplants, it all boils down to what's happening at the level of the MHC. So let's say that Ashley decided to donate a lobe of her liver to Matt, right? So she donates a lobe of her liver to Matt because Matt is dying from fulminant liver failure and he needs a new liver. So now you put the Ashley cell into Matt. Well, Matt's white blood cells are going to recognize Matt markers, but they're not going to recognize Ashley markers. So when they come into contact with this Ashley marker, these white blood cells are going to be triggered. They're going to recognize this as being part of non-self, and they're going to start destroying these liver cells, right? Because they don't recognize the marker on them. And that's a major problem in organ transplantation, is that the recipient of organs often reject those organs because the immune system launches a massive attack against that organ. So what can you do about that? Well, there are very interesting projects in the pipeline with respect to tissue engineering, but at the moment, the medical standard, you can never change the genetic marker in the surface of the cells. That will always be Ashley markers. The liver cells that divide to produce new liver cells will have Ashley markers on them. They're genetically determined proteins in the surface of the cell. You can't change the MHC markers. But what you can do is you can administer immunosuppressants, right? Chemical compounds, medications that suppress the immune system. So things like methotrexate, or there's a constellation of immunosuppressants ranging from steroids to chemotherapeutics, right? But these medications are going to essentially destroy your body's ability to make white blood cells. And if those white blood cells aren't around to trigger a rejection reaction, right, then that organ will remain in the body. The problem with that is if you suppress somebody's immune system, you open them up to a variety of opportunistic infections, and that's why the recipients of organ transplants have to be so careful about getting infections. An infection that you would fight in 2 days and hardly notice may kill somebody who's the recipient of an organ transplant. So that's the physiological basis of organ rejection. It just has to do with these MHC markers. If the MHC marker is in the place where it's supposed to be and these white blood cells come around and recognize it itself, they're not going to do anything. But if they don't, right, if they recognize it as being part of non-self, for example, if you have a transplanted cell and it's not the appropriate MHC marker in there, these white blood cells are going to trigger a reaction. Hey there, you've got a funny looking MHC. Are you from this part of town? Ha ha ha. So, yeah. Now we understand the structure of the membrane, right? So we understand the structure of cell membranes, we understand the role of phospholipids, cholesterol, and we understand what I mean when I say proteins are functional components of the cell membrane. Now we're going to talk about membrane permeability. Cell membranes, specifically in this case the plasma membrane, are called selectively permeable membranes because they allow some stuff across but not others. The material that's allowed across the membrane is referred to as permeable. You can also refer to permeable as penetrant, right? So permeable or penetrant particles can get just right across the cell membrane, specifically like the plasma membrane, no problem. Impermeable particles can also be referred to as non-penetrant. So you have permeable or penetrant molecules and you have impermeable or non-penetrant molecules. And impermeable or non-penetrant molecules can't move readily across the plasma membrane. So how do you distinguish between a non-penetrant versus a penetrant molecule? So non-penetrant / impermeable molecules can't move directly across the phospholipid bilayer. How do you determine, looking at a molecule, whether it's non-penetrant? Anything that's water soluble is going to be non-penetrant, right? So salts are non-penetrant, sugars are non-penetrant, amino acids are non-penetrant. Anything that's polar, right? AKA hydrophilic, AKA water soluble, AKA lipid insoluble, AKA charged, right? Anything that's polar is functionally non-penetrant. So things like sodium, chloride, water, calcium, hydroxide, hydrogen ions, glucose, amino acids, all of these things that dissolve in water carry a charge. And because of that, they are functionally non-penetrant. So they are non-penetrant solutes. They don't get directly across that phospholipid bilayer. The only way a molecule that's polar can become penetrant, right, can become functionally penetrant, is if you have a transporter or a channel to move that particle across the membrane. Otherwise, they are considered non-penetrant because they can't get directly across the phospholipid bilayer. Penetrant, aka permeable molecules, anything that's relatively small and non-polar, can get right across the phospholipid bilayer. The cell membrane presents no barrier to it. So remember, nonpolar slash hydrophobic slash water insoluble slash lipid soluble slash non-charged, they all mean the same thing, right, when we're talking about membrane permeability. So oxygen is a small nonpolar molecule. It can move right across cell membranes. Carbon dioxide, small nonpolar molecule, moves right across cell membranes. Most lipids, such as steroids, testosterone, estrogen, progesterone, move right across the cell membrane. So the importance of this is if you hear like lipid soluble or you read lipid soluble on a chemical label, what that really means is that your skin doesn't present any barrier to that molecule. You can rub that molecule on your skin and it'll go directly across those membranes that make up the cells of your skin and it'll get right into your bloodstream. So now, what we're going to talk about is diffusion. So we understand non-penetrant versus penetrant, meaning what solute particles can get directly across the phospholipid bilayer and what can't. And when you think about diffusion, diffusion is really the movement of solute particles, right, or any particle, but we're talking about solutes and solution chemistry now. Diffusion is the movement of solute particles from areas of high to areas of low concentration. So if you drop this purple sugar molecule into water, as that sugar dissolves, those sugar particles are going to move from areas of high concentration to areas of lower concentration. And they're going to continue to move if no force intervenes with that movement, right, from areas of high concentration to areas of low concentration until some sort of equilibrium is reached, until those particles are equally distributed throughout the solution. Then there will be no concentration gradient and they'll just all be kind of floating around. So diffusion is the movement of solute particles from areas of high concentration to areas of low concentration. And we refer to this movement as following a concentration gradient. So when solute particles move from areas of high to areas of low concentration, we say they're following their concentration gradient. So when you think about this with respect to cell transport, do you think this particle here will be penetrant or non-penetrant? So this is a cholesterol molecule. We know that it's lipid soluble and it's relatively small. So cholesterol forms the basis of steroids. It's lipid soluble, and because of that, right, because it's nonpolar or hydrophobic or lipid soluble, it's going to be able to interact with these nonpolar tails, right, because that's what accounts for most of the phospholipid bilayer, is that layer of fat in the middle. And these particles are going to be able to move right across the phospholipid bilayer. They don't need a transporter. They can just wiggle right through that phospholipid bilayer. Sodium, on the other hand, even though it's much smaller, and if I was to draw this to scale, sodium would look like a speck comparative to that cholesterol molecule, but I've drawn it big so you guys can see it. All you need to know that sodium won't get directly across the phospholipid bilayer is that sodium carries a charge. It is therefore polar, hydrophilic, lipid insoluble, water soluble. Polar or hydrophilic molecules cannot get directly across the phospholipid bilayer, so they will bounce off. We refer to these as being non-penetrant. So you have penetrant and non-penetrant. Do they bounce off the phospholipid bilayer? Then they're non-penetrant. The most important non-penetrant solute for you to really understand in anatomy and physiology is sodium chloride, salt, right? So sodium chloride is your most important non-penetrant solute. Now, passive transport mechanisms are transport mechanisms that do not require metabolic energy in the form of ATP. It relies on the kinetic energy held within concentration gradients. So when solute particles have a differential distribution, meaning that there is high concentrations of solute in one area and low concentrations of solute in another area, solute particles will move from areas of high to low concentration just based on their concentration gradient. So if a concentration gradient exists, there's no reason to input metabolic energy in the form of ATP because those solute particles are going to move in that direction regardless. So passive transport processes, which is the only type of transport we're talking about in this particular presentation, do not require metabolic energy in the form of ATP, right? They rely on the energy inherent in concentration gradients. So we don't input energy into them, right? Those solute particles just flow based on the energy held within concentration gradients. Now there are three types of passive transport processes you need to be familiar with. Simple diffusion. Simple diffusion takes place, first off, how do you know this is passive transport? Well, this solute particle is moving from areas of high to areas of low concentration. If you see solute particles are moving from areas of high to areas of low concentration, right, meaning moving with their concentration gradient, that is a passive transport process. Because those solute there's no reason to input energy in the form of ATP. You don't see ATP being used here by any of these molecules. That's because these solute particles will move with their concentration gradient. So these solute particles move from areas of high to areas of low concentration, and that's how we know it's a passive transport process. We know it's simple diffusion because these solute particles are moving directly across the phospholipid bilayer. And the only type of solute particle that can move directly across the phospholipid bilayer is a penetrant solute. Penetrant solutes, if I was to ask you, what do you know for a fact about these solute particles? Well, because they're penetrant and they can get right across the phospholipid bilayer, you know they're nonpolar. They have to be nonpolar, aka hydrophobic, right? And that's how they interact with these nonpolar or hydrophobic fatty acid tails, which account for most of the bilayer. That's why nonpolar solutes can wiggle across cell membranes. So when you look at this, you have your nonpolar solute, aka your hydrophobic solute, aka your lipid-soluble solute. Now, when you think about why this is important with respect to medicine and anatomy and physiology, let's say that a woman who's going through menopause is prescribed an estrogen cream. And that estrogen cream she rubs on her skin. But she rubs that estrogen cream on her skin, but those estrogen molecules end up getting into her blood and distributing around the body and doing what estrogen does. How do those molecules get from being on the surface of the skin into the bloodstream? Well, the only explanation for that is one, those molecules are penetrant because they're getting across the cell membranes that make up the cells of your skin, right? Two, if they're penetrant, estrogen molecules have to be nonpolar, aka hydrophobic, aka lipid soluble, right? So if you see lipid soluble on something like a chemical label, it means that substance is going to get right across those cell membranes. Your skin presents no barrier. So there are lots of medications that come in the form of creams, and if you're administering those medications on people, you should wear gloves. In other words, if you see anything on a chemical label that says lipid soluble, it means your skin doesn't present any barrier to it. It can get right across your skin into your bloodstream, so you should always wear gloves when you're working with lipid soluble substances. Channel-mediated facilitated diffusion is another type of passive transport. Now notice that in this textbook it said small lipid-insoluble molecules or solutes. When you look at these solute particles, they're small polar molecules. They are small polar molecules. And small polar molecules like sodium ions, potassium ions, calcium ions, chloride ions, water, get across the cell membrane utilizing channels. Remember that channels are specific. So if you have a sodium channel, that's only going to allow sodium across. If you have a potassium channel, it's only going to allow potassium across. So you see these channels and you go, okay, so this is a passive transport process because these particles are going from areas of high to areas of low concentration, right? So you're not going to need energy in the form of ATP because there's a concentration gradient there. It's channel-mediated facilitated diffusion because you have these little proteins here called channel proteins that are like little molecular donuts. And these particles have to be small polar molecules, small charged particles, notably ions. That's what really moves through channels. Now you have all sorts of different types of channels that you talk about in A&P, but what you're looking at here, right, is the firing of an electrical signal in a cell and the change in the electrical properties of a cell. And you're going to talk a lot about that when you get into classes like A&P. And what you're really talking about there is the movement of sodium and potassium through channels. And these channels open and close, and they allow sodium and potassium to move. And that's what accounts for a lot of the electrical characteristics of cells like neurons. You're going to be talking about channel proteins a lot, in other words. Then you have protein carrier facilitated diffusion, which is our final transport process. So you have simple diffusion, channel-mediated facilitated diffusion, and protein carrier-mediated facilitated diffusion. Notice that this carrier is large. It doesn't exist in just an open state. It needs to bind to its solute particle, change its shape, and kick that solute particle into the cell. Carrier proteins, like channel proteins, have specific solutes that they transport. So if you say a glucose carrier, you're talking about a molecule that just moves glucose. If you talk about an amino acid carrier, you're talking about a protein that just moves amino acids. And remember, this is a protein. This is just a blob model of a protein. Proteins are like little cellular machines. They do everything. So think about your handout and think about how you would characterize those transport processes. All of them are passive transport because there's a concentration gradient. You have your simple diffusion, your channel-mediated facilitated diffusion, and your carrier-mediated facilitated diffusion, and what types of molecules undergo those transport processes. What can you say about those molecules? The final type of transport process we're going to get into, which is another type of passive transport, is osmosis. Osmosis is a specialized term for the diffusion of water. So osmosis is a specialized term for the diffusion of water. Water, like every other particle in the universe, will move from areas of high to areas of low. Now, when we talk about the concentration gradient for water, we don't use the term concentration gradient because water has special properties. But the concentration gradient for water is called an osmotic gradient. So the movement of water from areas of high to areas of low water is called osmosis. And the gradient that exists for water isn't called a concentration gradient, it's called an osmotic gradient. Now, water, because it's a polar molecule, needs to move through the channel proteins. And there are a special type of channel protein that allows for the movement of water. They're called aquaporins. Every cell in your body, with the exception of one population of cells in the kidneys, are richly perfused with aquaporins, meaning that water can move across the membrane of cells pretty readily because every cell in your body has these aquaporin proteins. So, you have aquaporins, they allow for the movement of water. A specialized term for the diffusion of water is osmosis. The concentration gradient for water, we don't call it a concentration gradient, we call it an osmotic gradient. Now, when you think about the movement of water, you can think water follows salt and sugar, or water always follows non-penetrant solute. So water will always follow salt and sugar. Water follows non-penetrant solute. In other words, where there's higher concentrations of non-penetrant solute, water will move that way if it's allowed to move across a membrane. So water moves from areas of high water potential to low water potential, and it follows what's called an osmotic gradient. So let's look over here. You have water molecules, you have sugar molecules. Over here you have 32 water molecules, over here you have 18 water molecules. So before this process occurs, right, you have this membrane that's permeable to water but not to sugar. That's important, right? So it's permeable to water and not to sugar. Now in the beginning, when it resets itself, you have 32 water molecules over here and only 18 water molecules over here. The reason that there are more water molecules on the left than there are on the right is because there are sugar molecules taking up that space that water would fill. Sugar is a non-penetrant solute. So those water molecules are going to want to go from areas of high water, or the forces that promote the movement of water are going to move those water molecules from areas of high water to areas of low water. Areas of high water we say has high water potential. Areas of low water have low water potential, and water is moving down its osmotic gradient from areas of high water potential to areas of low water potential. Water will always follow non-penetrant solutes. Salts and sugars are always non-penetrant solutes. The reason why is water wants to dilute, right? It wants to move from areas of high water potential to areas of low water potential until an equilibrium is reached. Now, we refer to the gradient that promotes the movement of water not as a concentration gradient, but as an osmotic gradient. So if I asked you in which direction is the osmotic gradient targeted or directed before the movement of water, it would be directed from left to right, right? And then those water molecules move until an equilibrium is reached, and they create what's called an osmotic pressure. The region with the higher concentration of solute has the higher osmotic pressure, and osmotic pressure sucks water toward it. So you can create osmotic pressure by adding solute to a solution, and that will suck water over in that direction, creating a pressure that you can physically measure called an osmotic pressure. Now, this all feeds into a concept that's really important called tonicity. So, tonicity refers to what a solution will do to the size and shape of a cell. It's not interchangeable, and Khan Academy kind of gets this wrong, and I love Khan Academy, but they don't do this one well. A solution has a tonicity comparative to a cell. A cell does not have a tonicity comparative to a solution, because by definition, tonicity refers to what a solution will do to the size and shape of a cell that's put in that solution. So the cell can't do anything to the size and shape of the solution, but the solution can certainly influence the size and shape of a cell. So we say solutions have certain tonicities. Any solution that's hypertonic will cause water to be sucked out of the cell. When water gets sucked out of the cell, these cells shrink and shrivel and undergo a process called crenation, Crenation. So these cells, right, that are placed into a hypertonic solution, that solution causes water to be sucked out, and those cells shrink and shrivel or undergo a process called crenation. When you put cells into an isotonic solution, there's no net movement of water into or out of the cell, right? Meaning that water doesn't flood into the cell or get sucked out of the cell. It's moving at an equal rate, so the cell doesn't change its size or shape, and we refer to that as being an isotonic solution. Any solution in which you place a cell that causes water to rush into the cell, causing the cell to swell and potentially burst, is called a hypotonic solution. So if I ask you to characterize a solution with respect to tonicity, I'm asking, is it hypertonic, isotonic, or hypotonic? Now, one of the things that dictate tonicity, the only type of solute particle that can influence tonicity is a non-penetrant solute. So a salt or a sugar. Those are the only types of solutes that can influence tonicity. Penetrant solutes don't, for reasons that you discuss in AMP1. So when you look at this and you think about IV science, different IVs are run under different scenarios or for different reasons. So here we have an IV that says 0.9% sodium chloride. And this IV, you're going to connect to a machine that's actually, you're going to insert a syringe into somebody's vein, and you're going to run that IV, this solution, slowly into their blood. So you're going to want to make sure to get that right. There isn't just one type of IV solution that you run on people. You run different types of IVs depending on what you want the solution to do. So when you think about a solution that has 0.9% sodium chloride, the inside of the cell has a concentration of 0.9% sodium chloride. Sodium chloride is non-penetrant, so wherever it is, it's going to stay, right? It doesn't get into or out of the cell because it bounces off that plasma membrane. Now, when you're wondering about tonicity, know that all the cells in your body have aquaporins. So if you create an osmotic gradient, that water will move along that osmotic gradient. Now, another way of saying that is water follows salt and sugar, or water follows non-penetrant solute. So when you're looking at this and you put a cell into a 0.9% sodium chloride solution, think about the percent water. So one, is there a higher concentration of salt outside the cell than inside the cell, or in the ECF comparative to the ICF? The answer to that is no. The concentration of salt in the ECF and the ICF is the same. There's nothing for water to follow, so you're going to get no net movement of water. Another way to look at this is if the solution is 0.9% sodium chloride, it's 99.1% water. If the inside of the cell is 0.9% sodium chloride, it's 99.1% water. 99.1% water inside and outside the cell. There's no osmotic gradient to promote the movement of water. So water is not going to go anywhere, and we refer to these as being isotonic solutions. You run isotonic solutions when you want to increase somebody's blood pressure, for example, if they're hemorrhaging. You also run them in certain types of dehydration, right? Not all types, but certain types of dehydration. And you run them during things like surgery just to increase blood volume without playing around with the size or the shape of a cell, right? So they play a really important role, these particular solutions, in treating things like certain types of dehydration and low blood pressure that can be caused for a variety of reasons. You don't want to, if you're doing that, you don't want to play around with the size and the shape of the cells. You don't want water flooding into your cells or being sucked out of your cells. But that's not always the case. Sometimes there are solutions that you want to run under certain scenarios. So if we look here, we have a 0.45% sodium chloride solution, right? By definition, a 0.45% sodium chloride solution would be a hypotonic solution. And let's say that I asked you a question on the exam like, okay, Spencer goes to an event called Burning Man in the desert. And at this event, he's wearing a pair of cut-off jean shorts and a macrame shirt. And he gets lost in the desert. And he doesn't have water for 48 hours, so he gets so dehydrated that his cells start losing water. Well, if your body's cells start losing water, it's kind of like wilting of a lettuce leaf. And what do you want to do with a wilted lettuce leaf? Well, sometimes you put those wilted lettuce leaves into pure water and it plumps that lettuce back up. And we're doing a similar thing when we run a hypotonic solution on somebody. We're running a solution that we know is going to cause water to move from the outside to the inside of the cell, or from the ECF to the ICF, and we're pumping water back into the cell, right? So you use hypotonic solutions when you want to pump water back into the cell, for example, in the case of severe intracellular dehydration that could be caused by being in the desert for a prolonged period of time. You also run hypotonic solutions when you want to dilute your blood. So for example, if a kid eats a bunch of salt, right, and that happens sometimes, like just eats a bunch of salt and throws a bunch of solutes in his blood plasma and increases the concentration of their blood plasma, you want to dilute it back down. So when you think about a question like this, I want you drawing things out. You go, okay, so does water move from the ECF to the ICF, ICF to the ECF, or is there going to be no net movement of water? That's the first thing you ask yourself, because that's going to determine whether your solution is hypotonic, right, hypertonic, or isotonic. So now you draw it out. You draw a little cell system and you go, here's the ECF and it's 0.45% sodium chloride. Here's the ICF, which is 0.9% sodium chloride. Now, water always follows non-penetrant solutes, specifically salts and sugars. Our most important non-penetrant solute is the salt sodium chloride. So where is there a higher concentration of sodium chloride? There's a higher concentration of sodium chloride in the cell. Therefore, the water will move from the outside of the cell, the ECF, to the inside of the cell, the ICF, because water follows salt, right? So that water will move from the ECF to the ICF, the cell will swell, and if it's hypotonic enough, 0.45% isn't hypotonic enough to cause the cell to lyse or burst open, but that cell will plump up, right? Water will move into that cell and that cell will plump up. And because you got net water movement into the cell, that solution by definition is hypotonic. Another way to look at this is if the solution has 0.45% sodium chloride, then that solution would be 99.55% water. And inside the cell, if it's 0.9% sodium chloride, that solution would be 99.1% water. 99.55 is higher than 99.1, so water will move from areas of high water to areas of low water down its osmotic gradient. Water follows non-penetrant solutes, or water moves from areas of high water potential to areas of low water potential along an osmotic gradient. It's two ways of thinking about the movement of water. So you have hypotonic solutions, and under what circumstance would you move those? Or hypotonic solutions, and under what circumstance would you use those? Finally, we have hypertonic solutions. So look here at this solution that you're going to be running on somebody. It's half a liter and it's 3% sodium chloride. So draw out your system. Draw your cell and go, okay, inside of a cell, 0.9% sodium chloride. Outside of the cell, 3% sodium chloride. Water follows salt. Water will move from the ICF to the ECF, right? Meaning that cell, it'll cause cells to lose water and shrink. So under what circumstances would you run something like a hypertonic solution? Well, let's say you were suffering a condition of hyponatremia, meaning that the sodium concentrations in your blood went way, way down due to whatever fact. It could be because you're taking an antidepressant. Hyponatremia is a really common side effect of antipsychotics and antidepressants. That's why you always ask your patient what medication they're on, right? Or you've been running and you've been drinking distilled water, but you haven't been, getting your corresponding electrolytes, and not a little run, not a couple miles, but maybe you've been out all day in the sun and you've been doing that all day. Your sodium levels can drop. So if you want to bring sodium levels back up, it makes a lot of sense to run a hypertonic solution because you want to suck water out of those cells that have been plumping up. You don't want those cells bursting on you. Another situation would be something like edema. So here we have what's called cerebral edema. Edema is the accumulation of water in tissues. And if you want to get rid of water in tissues, for example, if there's an accumulation of too much water in the brain, too much cerebrospinal fluid, and you're like, oh man, we got to suck some of that water out. Hypertonic solutions, you can't go in there with like a vacuum and go, right? But you can run a hypertonic solution, and that hypertonic solution could suck that water out. So think about those things. And the last thing I'm going to show you is the first. So every lab that you have, like in A&P1, one of the labs that my students have the most trouble with is the passive transport lab. It's a conceptually dense lab. And it's because coming into the class, you make the assumption that they understand things like osmosis and tonicity, and then when you do this lab, you see that, oh, there are some major content gaps, and I personally go back and kind of cover those. Some instructors do, some instructors don't. But when you look through this lab, one, it's telling you how to convert molarity into osmolarity. So it's walking you through that. What's the relationship between osmosis and osmolarity? Osmolarity is the truest measure of concentration. And notice that when they're asking you these pre-lab questions in this lab, they're saying ECF 300 milliosmolarity, ICF 300 milliosmolarity. Those are units of concentration. And they're just asking you, do you understand the basics of how water moves? So will osmosis occur? That's kind of a dumb question. I didn't write the lab. Osmosis is always happening. But will you get net movement of water? The answer is no. Right? Water won't be moving into the cell or out of the cell at any faster rate, so it's not going to change the size and shape of the cell. So in which direction? Not applicable. With a cell volume change? No. And then you go and you look at this, 250 milliosmolarity comparative to 300, and you get these units of concentration, and you have to predict, is osmosis going to occur or is osmosis not going to occur? And that fundamental understanding leads into doing our first real wet lab that involves blood and gloves and following safety protocols and following rigid lab protocols. And if you're too bogged down with just understanding the basics of the lab, then when you get into the more complex topics that come up with respect to the solution and how you can tell the solution is hyper or iso or hypotonic, it becomes problematic. So having these fundamentals is important. And that's it.